Hello and welcome. In this video, we'll be talking about the physiology of thyroid gland. It will cover three main parts. First, the synthesis and secretion of thyroid hormones. Next, the function of these hormones. And third, the regulation of its secretion. Let us first discuss the synthesis and secretion of thyroid hormones. To understand this, let us first take a look at the physiological anatomy of the gland. It consists of follicles wherein each thyroid follicle is lined by cuboidal epithelial cells and filled with colloid, the major component of which is thyroglobulin. This glycoprotein acts as both a scaffold for hormone synthesis and a storage form. Between the follicles, parafollicular cells secrete calcitonin, and the gland is richly supplied with blood, about 5 times its weight per minute. The thyroid secretes mainly thyroxine, T4, about 93%, and triiodothyronine, T3, about 7%, for the formation of which iodine is indispensable. Although secreted in smaller amounts, T3 is more active and potent. Our body requires about 1 milligram per week of iodine for normal hormone formation, usually supplied by the diet, and deficiency is prevented by the iodinization of table salt. Let us now zoom in on an epithelial cell to understand the steps involved in the formation of these hormones. Ingested iodide is absorbed from the gastrointestinal tract and enters the circulation, about 1/5 of which is trapped by the thyroid for hormone synthesis and the rest is excreted in urine. The first step in hormone synthesis is iodide trapping, also called the iodide pump. The basal membrane of thyroid cells contains the sodium iodide symporter, which actively transports iodide into the cell, driven by the sodium potassium ATPase. Now, this also helps to keep a low sodium concentration within the cell, thereby facilitating the sodium iodide symporter. This concentrates iodide up to 30 times the plasma level and under maximal stimulation up to 250 times. Iodide then passes into the follicular lumen through a transporter called pendrin, which transports iodide against the chloride ion. Meanwhile, the thyroid cell synthesizes thyroglobulin in the rough endoplasmic reticulum and the Golgi apparatus and secretes it into the follicular lumen. Each thyroglobulin molecule contains about 70 tyrosine residues, which serves as the substrate for iodination. Within the lumen, the iodine becomes associated with the tyrosine residues of thyroglobulin, giving rise to monoiodotyrosine (MIT) when one iodine attaches and diiodotyrosine (DIT) when two attach. This step is known as organification. But before this happens, iodide needs to be oxidized to iodine, as oxidation stabilizes the bond between iodine and tyrosine residues within thyroglobulin. This is possible by the enzyme thyroid peroxidase in the presence of hydrogen peroxide. This is then followed by organification, which we previously discussed. Subsequently, a process of coupling occurs when one monoiodotyrosine combines with a diiodotyrosine. It forms T3, whereas when two diiodotyrosine residues couple, they form thyroxine. Small amounts of reverse T3 are also formed, but this is physiologically inactive. Thus, the follicular colloid contains large amounts of iodinated thyroglobulin with embedded MIT, DIT, T3, and T4. This forms a vast storage pool capable of supplying the body's needs for 2 to 3 months. Clinically, this storage explains why hypothyroid symptoms often appear only gradually after iodine deprivation. When secretion is required, droplets of colloid are endocytosed, fuse with lysosomes, and undergo proteolysis, liberating T3 and T4, which later on diffuse into the blood. While unused monoiodotyrosine and diiodotyrosine are deiodinated intracellularly and the iodine is recycled. Congenital deficiency of this deiodinase enzyme may lead to iodine deficiency due to deficient intracellular recycling. Occasionally, thyroglobulin itself may be secreted into the circulation bound to a protein receptor called megalin. This is a minor pathway, and in the blood, more than 99% of thyroid hormones are bound to plasma proteins, chiefly thyroxine binding globulin, with smaller amounts bound to transthyretin, also known as thyroid binding prealbumin, and albumin. All of these are synthesized in the liver, and this ensures a slow release to the tissues. Half-life of T4 is about 6 days, whereas T3 is about a day. The mechanism of action of thyroid hormones is primarily genomic. Within the tissues, a substantial proportion of T4 is converted to T3, the more active form, which then enters the nucleus, binds to the thyroid hormone receptors, subsequently forming heterodimers with retinoid X receptor and activating DNA response elements. This leads to transcription of many genes and synthesis of numerous proteins, particularly metabolic enzymes. Clinically, this genomic action explains the slow onset but prolonged effect of thyroid hormone. In addition to these genomic effects, thyroid hormones also exert non-genomic actions, including direct effects on mitochondria, regulation of ion channels, and modulation of intracellular signaling pathway, which contribute to their rapid cardiovascular and metabolic responses. Now let us consider the functions of these hormones. They are the principal regulators of the basal metabolic rate and also cause changes in body weight. They heighten excitability and accelerate thought processes. Excess leads to nervousness, reduced sleep, and tremor, while deficiency causes sluggishness and somnolence. Respiratory rate is increased as they increase oxygen consumption and carbon dioxide production due to elevated BMR, leading to enhanced ventilation. These hormones also increase heart rate, cardiac output, and contractility, primarily due to increased oxygen demand and heightened sensitivity to catecholamines. In hyperthyroidism, persistent overstimulation weakens the myocardium and may cause heart failure. Vasodilatation also occurs, which is due to excess heat production. In the GIT, they increase motility and secretion, and hence, clinically, diarrhea occurs in hyperthyroidism, and constipation is typical of hypothyroidism. In muscles, prolonged excess can cause weakness due to protein catabolism, and hypothyroidism results in slow contraction and delayed relaxation. They increase carbohydrate absorption from the intestine, enhance glycolysis and gluconeogenesis, and stimulate fat mobilization. Plasma cholesterol, triglyceride, and phospholipids are reduced. Clinically, hypothyroidism produces hypercholesterolemia, whereas hyperthyroidism lowers the cholesterol levels. In women, both hypo and hyperthyroidism cause menstrual disturbances, and in men, libido may fall. They're essential for growth and development, especially of the brain and skeleton during fetal and neonatal life. Clinically, congenital hypothyroidism, if untreated, results in cretinism with dwarfism and permanent mental deficiency. Excess thyroid hormones, although lead to increased height initially, may cause early epiphyseal closure and thereby reduced overall height. Finally, let us discuss the regulation of thyroid hormone secretion. The principal regulator is thyroid stimulating hormone, secreted from the anterior pituitary. TSH stimulates iodide trapping, thyroglobulin synthesis, iodination, coupling, proteolysis, secretion of T3, T4, and hypertrophy of thyroid cells. The fastest action of TSH is the stimulation of proteolysis of thyroglobulin, leading to the release of preformed T3 and T4 within about 30 minutes of TSH elevation. These effects are mediated through the cyclic AMP second messenger system. The secretion of TSH is in turn controlled by thyrotropin-releasing hormone from the hypothalamus, which acts via the IP3 calcium pathway. There is a strong negative feedback control. Elevated T3 and T4 levels suppress both TSH at the pituitary and thyrotropin-releasing hormone at the hypothalamus. Thank you so much. Hope this video was useful. Please like, share, comment, and subscribe.